Hello, I'm Robert Walker, and this is my presentation for a paper that I plan to submit to astrobiological journals in spring 2021. NASA's Perseverance rover is on its way to Mars as I speak. The plan is to drill and cache samples on Mars. ESA and NASA plan to return these samples unsterilized to Earth by 2031. So this graphic shows the timeline. You can see there, that's the mission. It's now called Perseverance, but it's on its way to Mars. And then it'll land in spring 2021. And then the two follow-up missions, there's the ESA sample retrieval lander, which will go down to the surface and go and retrieve the sample, and that also has the Mars Ascent vehicle with it, and that will take off into orbit. And then there's the Earth Return Orbiter, which will go and meet that. This is meant to be above Mars' surface, and they'll take it all the way back to Earth. And then this is the sample receiving facility, which they plan to complete by 2031. However, the legal process hasn't yet been started, and nor has the build of the facility been started. And estimate in the planetary protection literature, its legal process will take at least 8 or 9 years. It'd be by far the most complex thing that NASA's ever had to do for a space mission. And it would involve NASA and then also ESA as a partner, and then that involves the United States, the EU, United Kingdom, Canada, legal processes in all these places, and also international legal processes, international treaties, and organisations such as the World Health Organization will get involved, and anything to do with agriculture and fisheries and such like in all these countries. And it's really quite ambitious to suppose that the process in the United States alone would take 8 to 9 years very streamlined, and it could easily be extended well beyond that. So it seems quite ambitious, but maybe possible to finish that starting in 2021, they haven't started yet, and finish it in 2031. Then when you come to the facility, that takes at least 11 years to build the sample receiving facility. So that would mean you can't quite do it by 2031. However, if you think of these both being done concurrently at the same time, then it would seem that we could finish it and the sample could be returned sometime in the early 2030s, perhaps, if there are no serious obstacles. However, I'm going to show in this paper that actually the legal process and the sample build have to be concurrent, have to be consecutive, one after the other. And what's more, we don't yet have the technology to start the build. And so if that is the situation, then really 2040 is very optimistic. It could easily be into the 2050s before it could be returned. And that's just far too late. We know much more about Mars by other methods, but if it's going to take that long. So that basically is the outline of this paper, and there's lots more to it. And then, so what I think is going to happen, the two possibilities: either NASA sterilize the sample, or they return it to somewhere safe, such as above, I suggest, in the Earth's ring plane, essentially, if we had rings. The Laplace plane, slightly inclined to the geostationary Earth orbit, and maybe 10,000 kilometers above that. I figure that out as being the safest place of all. There's some papers I'm using as a source there. Anyway, so I'll get to that later, maybe not in this video. This video I'm going to talk about the very basic matter of the timeline. So this paper is accessible to multiple disciplines as for the legal process. And if you're expert on planetary protection, please bear with me for a few minutes, because I'll describe the basic issues to everyone else. If you haven't followed recent planetary protection papers, you might well think there's plenty of time. After all, Kennedy made his speech in 1962, and the samples were returned by 1969, so you would think 7 years, so there's plenty of time between now and 2031. But the Apollo mission is now thought as most valuable, so lesson for how not to do a sample return. And this is what I'm using my source when biospheres collide. And I'll just give you an idea of how it's seen nowadays. Actually, when the Apollo astronauts and one of the NASA frogmen opened the door for the capsule, and then he threw in the biological isolation garments into the astronauts. At that moment, then the astronauts reported that the inside smelled like gunpowder, and there's very, very fine dust from on the moon, and that was everywhere inside. It would have got into the ocean at that point, there's no doubt about it. And there was an earlier point when it was descending and they vented air from the capsule, it would have got into the ocean at that point too. And then this seems very kind of archaic, sort of Middle Ages kind of thing, that when to modern biology, that astrobiology, that they then, when the astronauts got out and they would have had dust on the outside of their biotech suits, which they put on inside the Apollo capsule. So they then swabbed the astronauts, all the outside of their suits, their legs and so on, and their feet and everything, with a solution of, now what was it again? I've got sodium hypochlorite, I think. Oh, it's up here, maybe. Sodium hypochlorite. And they threw the swabs into the ocean. And with our modern understanding of polyextremophiles and the limits of size of an organism, that seems, it really seems very naive today. And of course we know there never was any risk, but they didn't know that back then, because Carl Sagan had published a paper suggesting there could be liquid water suitable for life just a few meters below the lunar surface. And they eventually found that his model wasn't quite correct. But at the time, it seemed quite possible, even with the near vacuum atmosphere. So the original plan was to hoist the capsule out of the sea with a crane, but this was changed as an executive decision, and the mission planners were worried that astronauts would get seasick, bobbing in a small capsule waiting for the crane to lift them off. So that's why they did that. And there are many other executive decisions and lapses, including an expert was given access to the samples without any training, and then there was a small cut found in one of the gloves, which led to 11 technicians having to go into quarantine. And then Carl Sagan observed, and so that would be needed for a sample receiving facility, Carl Sagan observed that the incubation period of leprosy is multiple decades. And then if any of the astronauts or technicians developed serious illness during quarantine, the plan was to immediately remove them to hospital. So the quarantine procedure gave no protection in the very situation where it's most needed. So, and this is not just a problem for the NASA astronauts. If you have technicians with a Mars sample receiving facility, they could be in quarantine, and then the same thing applies to them. So you've got to figure out the quarantine rules. That's part of the legal process. The guidelines, NASA guidelines, were published on the day of launch of Apollo 11 without peer review, and NASA didn't have legal authority to impose quarantine. They did it under the auspices of the Surgeon General, and the legality of this has never been tested, as there were no objections. So we don't actually have anything that's passed legal process to protect, to tell us how to do a sample return. And it's all done under the legally weak Outer Space Treaty, and modern environmental legislation didn't exist back then, and the guidelines have now been rescinded. So the modern legal process would be a completely different thing. They just couldn't do that published on date of launch. They couldn't do that sort of thing. So the modern legal process would take at least 8 or 9 years. I said separate. I explained that just up there with that graphic. So and NASA hasn't started on either of those. So this, and then as I researched the paper, I discovered that the technology requirements of the most recent sample return study by the ESF in 2012 they go beyond the capabilities of present-day technology for filters. This photograph shows the problem. And this is a bacteria that has passed through a 100 nanometer filter. And this happens when bacteria are in starvation mode. And they get very, very thin, basically, and then they can go through these tiny, tiny holes. And that was the discovery between 2009 and 2012. And they also found that that was very similar to the theoretical minimum diameter for terrestrial microbe. And HEPA and ALPA filters, such as they use in all the biosafety designs that I've seen, then they're not tested down to this size. And so if you go and look at what it is equivalent to the size of the SARS-CoV-2 virus for COVID-19. And there's the HEPA filters do protect the workers, the nurses and the doctors in intensive care units. But they do it by keeping out the larger droplets and then reducing the viral load they can't keep out individual viral particles. But our innate immune system is able to fight off a small viral load. So this experimental filter, this can filter out 88% of particles at 50 nanometers and larger, but that's not enough. If you go and look at their requirement, they said that you can't have a single particle at that size, 0.05 microns, not permitted to leave the facility. And this would certainly be looked at during a legal process, and this document will be brought up, and NASA will have to say, what you're going to do about that. And so far, there are no plans, there are no designs that are suitable that can actually do that, and there are no filters available. Technology doesn't exist. You could, of course, make tiny, tiny nanopores, but the problem is making that into a technology that can use as a filter in a practical way in a building or a biosafety cabinet. And they actually set a 1 in a million chance of 0.01 microns, and that is and below that is acceptable. That is based on the smallest gene transfer agents of 0.03 microns. This was another discovery between the two reviews, where they found that gene transfer agents which can transfer genetic capabilities to species as different as from, say, a fungus to an ant, and that was in the case of the genetic encoding for making carotene, then in this particular experiment they put a gene transfer agent for transferring antibiotic resistance into seawater, and it got transferred to totally unrelated forms of life very readily overnight in seawater. So if there was a gene transfer agent in the sample, it could easily get into the seawater just the next day. So the 2012 study, they set a limit of 1 in a million probability of release of a single particle at 0.01 microns. Now, the Europa's design is the only one that actually cites this study of the recent articles that I've looked at. Then on designing a Mars sample return facility, and they cite this as being a 1 in a million chance of an unsterilized particle greater than 0.1 microns. This seems to be a simple typo. If you look at the Europa ESF study, then it says a single particle of 0.01 microns should be 1 in a million, less than 1 in a million, and they put it as 0.1 microns. And this is like the foot and feet and meters thing with that spacecraft that got lost. It's just miscommunication. And then they have the release of a single unsterilized particle larger than 0.05 microns is not acceptable under any circumstances. And also this 1 in a million chance wasn't per particle, the way you normally measure it for filters. It was a 1 in a million chance of a single unsterilized particle into the Earth's environment over the entire lifetime of the facility, not per particle. And they said that can be reviewed, but this is unacceptable under any circumstances. That would need detailed review if you can't achieve that. Now, I argue in my paper that at 8 years later, they said that because of the startling reduction from 0.25 microns for the 2009 study to 0.05 microns, because of the discovery of these starvation-limited bacteria, then they said that there's, with such a dramatic change, then we need to do periodic review. They think they've got as far down as it'll go, but there could be new infectious particles and things that would mean it should be lower. So 8 years later, then it could easily lead to another dramatic reduction, in my view. And there's one, we've had big development in synthetic biology, and in the last 8 years, huge amount happened on that, and on early life, and things like epibionts, which live on the surface of organisms, and we use some of the organics produced by the organisms. They don't make them themselves. Anyway, with early life, then a starvation mode early life form, and of course Mars, sample from Mars, then that is a very nutrient limited, so it would be no surprise if some of the microbes in the sample were starvation limited in this way. So starvation limited early RNA form of life, it could be it could have only 5 genes, and the thing is that modern life, with the DNA, the RNA, the messenger RNA, the ribosomes, and it goes around in a circle, and you need all of them, and then you need the ribosome, you need to make the enzymes in order to catalyze the reproduction of DNA and the translation of DNA into messenger RNA, and there's just no way you could just build that up step by step. It has to have evolved from something simpler. So if you go back and look what a simpler form of life there could have been before the simplest form of modern terrestrial life, which is just all tied together in such a way that it couldn't just come together, then to start with, it would likely have ribozymes, which are much smaller than ribosomes. It would have only one informational biopolymer instead of 2. It would likely not have proteins, and this is the RNA world hypothesis, only one informational biopolymer RNA. The ribozymes are made of fragments of RNA stuck together in a kind of higgledy-piggledy looking kind of way, but it's actually quite structural. And so that's life made like that, then the very first exactly reproducing life, almost exactly, could be just 5 genes, according to the experts. And that could fit through, that could be down to, and they found that dimension of, they looked at the ALH 84001, that meteorite, which hit the news as being thought that originally thought it had Martian life in it, and now it's just ambiguous. Nobody knows if it's life or not, and it's safe to assume it isn't, because it would be a big discovery if it is. That there are abiotic ways of making the same structures. But they looked at, and the main issue was that it looks too small for terrestrial life, but it could be, RNA world life could look like that. So that, if that was accepted, and then this would need to be re-examined again with the last 8 years research to see if an RNA world cell could be that slender. If it could, the minimum requirement would not be 0.05, it would be more like 0.01. So that would bring the unacceptable range here right down to there, and all that would be red. That can't be ruled out. So we are in this situation where the technology doesn't exist for 0.05 microns. 0.5 microns mightn't be the final requirement. So in this situation, there's no way that NASA knows what it's going to build. That's why I explained all that. I thought it possible to explain that we have quite a few steps in front of us to find out what the minimum requirements are, and then to develop the technology, test it, know how it's going to be used in buildings. So you could add quite a few years to that 11 years, even without the issue of the legal process. However, NASA has a requirement that it can't start to build, so naturally NASA won't want to build something until they know what they're going to build. Now, you could imagine, okay, they'll spend a few years, they'll first of all do the review, they'll develop the new requirements, whatever they are, then they will build the technology, they will test it and everything, and maybe by the mid-2020s, they know what they're going to build, and sometime mid to late 2030s, then they will have their sample receiving facility. However, NASA has a requirement that they have to complete, well, first of all, naturally, they have to complete an enterprise architecture review, an enterprise service review for such an expensive building. And this is a legal requirement. If the proponents for this new NASA project will have to complete that before it can unlock the funding and start the proper build and the proper designs of the thing. So you might think, well, they could do that on the basis of once you've worked out the technology and you've got the Mars sample receiving facility. But the problem is that with the sample receiving sample study, then they have the remit. We've got to find a safe way to return it. So they look for the best available technology, and then they set out their requirements, and then they say this is what you've got to build. Now the problem is the legal process works in a different way. In the legal process, NASA is a proponent, and NASA have to prove that it's safe to do what they're doing, because the precautionary principle says that you have to do it, you have to prove that whatever you're doing is safe. It's not for the person who's examining it to prove it's unsafe. The person who's proposing it has to prove that it is safe, or the entity has to prove that it is safe. And so then it's NASA don't know in advance until they've completed the legal process that they are going to get approved. Their design they've put before everyone else, experts from many different disciplines, looking at it from many different angles and saying how does this protect the fisheries or whatever it is, and how are you going to deal with quarantine. And so quarantine of technicians. No, all these questions are going to come up, in many detailed questions are going to come up, and in the process the design may very well get changed. So NASA can't know in advance what they're going to build until they've completed the legal process. So that is why I say that the legal process has to come first. So, the design has to first. First of all, they have to complete the review, then they have to do the design. And they've got to have something that's at least reasonably plausible that this is going to be a working technology before they start the legal process. It'd be much more challenging to get it through the legal process if they don't know what they're going to build yet. And they have to try and converse and say, well, actually at the same time as we're doing the legal process, we are researching and we hope to have the technology finished soon. Then that will make the legal process obviously more complicated and more difficult. So that's why I think even 2040 is quite optimistic. If you add it all end on end, maybe 2050 would actually be more likely to complete all of this. So I don't think NASA are going to do this. Now, I don't think they're going to go through this legal process. I think what NASA are going to do is that they're going to either sterilize it or they're going to return it to somewhere else. So I'm going to talk about that in my next short video. But meanwhile, I'm just going to talk about one more thing here. And yes, for any experts listening, have I summarized your discipline correctly, and have I missed anything? Have I made any mistakes here? I've shared with this lots of people. Nobody so far has found any mistake. Do you see any mistake in what I've just explained? Is there something I'm missing? Is there some way that they will be able to do it much faster? So then I want to look at one other thing. I talked about how NASA, the proponent, and it's the precautionary principle. In the sample receiving studies, then they have looked at what would count as actually being quite small scale effects. Similarly to a biosecurity laboratory, a BSL for a laboratory, where the gold standard is that you've got a 1 in a million chance of release. Then that's regarded as being secure enough. And for something like anthrax, and then they look at the possibility if you have got a release, then then they're talking about a small-scale effect which they could then clean up. They could find everyone who's been infected by this agent and deal with it, quarantine them, isolate them, and so on. But Carl Sagan was especially concerned about large-scale effects, and this is not really looked at in the sample receiving facilities, not in detail. So he said, the likelihood that such pathogens exist is probably small, but we can't take even a small risk with 1 billion lives. Something that comes out of the receiving facility, he was thinking about affecting humans, but it could also affect the biosphere. And it is possible for a disease of a biofilm on Mars to infect the human lungs, which looks very much like would look very much like a Martian biofilm. So people who say, oh, you couldn't have pathogens, Legionnaire's disease does that. It's a disease of biofilms that affects our lungs. So when people say, oh, you can't have a pathogen from Mars, you can. But so the studies don't seem to consider such really large-scale effects. And and there's one particular thing, but it is a central question in synthetic biology, which takes the large-scale effects much more seriously than planetary protection literature does. In synthetic biology, so scientists have now mapped out methods to create mirror life in the laboratory, and that leads to discussions about releasing mirror life into the environment. And the worst case is it's not pretty. You don't want this to happen. Most terrestrial life would be unable to metabolize most mirror organics, such as the starches, proteins and fats, and that would give it a huge competitive advantage. And so you get these experts saying that they believe that it's possible that the mirror life would quickly consume all the available nutrients, nothing else would be able to compete with it, and then leaving very few or even no nutrients for normal life, which would eventually develop the isomerases. Indeed, there are some microbes in Antarctica which can eat mirror life already, but most terrestrial life would not be able to eat it. And then eventually, because if they're talking about the mirror life cyanobacteria that got into the environment, then blue-green algae, then it would cover the oceans, and then when it dies, nothing would eat it. It would fall to the bottom of the sea, and it would keep growing and falling, never cycle back. It would take lots of carbon dioxide out of the atmosphere, and then you have the C4 crops like corn and sugar cane. They would be all right, but all the other agricultural crops, they just wouldn't be able to survive once the carbon dioxide goes down. And eventually, even the corn and sugar cane, they might very well depend on the microbes in their soil. Once all the microbes in their soil are mirror life, they might have problems too, eventually, as it evolves over centuries. I think humans would survive in that situation. They seem rather pessimistic. I think humans would survive by doing paraterraforming, covering the surface of Earth's terrestrial surface with huge greenhouses. They would have to find a way to keep out mirror life, which would be a huge challenge. But I think they would eventually figure out how to do that, and humans would survive. But we don't want this situation to happen. Absolutely not. And this counts as being a very severe effect. It's not just quantitatively that you've got instead of a dozen people, you've got 100 people. You've got something qualitatively completely different, a permanent effect on the biosphere of Earth that severely affects the lives and livelihoods of billions of people way into the future, indefinitely. And to sort of prevent that needs something better than a biosafety laboratory. And we just can't do that. For the biosafety laboratory, their plan is to make sure that synthetic mirror life depends on chemicals not available outside the laboratory. But you can't do that for Martian life. So then, if we go back, we're talking about the precautionary principle. So the different versions of the precautionary principle, they all have this idea that the proponent has to prove that it's safe. But the different versions, there's the best available technology, which is what you use for what they were using for the sample return study for the like of a biosafety laboratory with this 1 in a million chance, and you just do the very best you can to contain it. However, there's another version called the prohibitory version of the precautionary principle, and in that case, if you can't prevent it, then you can't do the activity. If the effect is very severe, like the ones I've just outlined, then that would seem to be that during the legal process, then when discussing it, if there's someone like Carl Sagan there, he would certainly be arguing for the prohibitory version of the precautionary principle. Of course, he'd be desperately keen to return samples from Mars and study them, but he just values human life and the Earth's biosphere far higher than the extremely high scientific interest he would have. So that is something that can happen, that could happen in a legal process, that not through not wanting the sample to be returned, but through highly valuing Earth's biosphere. Then they may decide that we have to use the prohibitory version. Then in the European Space Foundation study in 2012, they looked at the prohibitory version and they said that we can't use the prohibitory version because if we did, then we wouldn't be able to return the sample, because we can't design it in such a way that there's no appreciable risk. They can make it small, but they can't have no appreciable risk of a sample breach. So if that is still the situation today, and especially bear in mind we don't even have the technology yet, but that could very well mean that we just simply can't return a sample from Mars unsterilized. So we need to remember that is a possible outcome of this process. And so that reinforces that NASA can't assume from the outset even that their proposal is going to be accepted at the end. Now, all this is because we don't know what we're returning. That's the reason for all this complexity. We would have to take exactly the same precautions to return a sample from a Mars-like planet orbiting Proxima Centauri, or even orbiting a star in the Andromeda Galaxy. Because we don't know, and despite what some people will say to you, we don't know that the Martian life is related to terrestrial life. Astrobiologists, when they're designing instruments to search for life on Mars, don't search for familiar life. They're well aware that what could be there could be something that is not like anything we've seen before, like for instance mirror life. It could be nobody quite knows why Earth hasn't got mirror life. It might be that on Mars both forms exist. And mirror life could have evolved, there's no reason for them to be identical to each other. There's no way to flip, unless there is something developed there to kind of flip organisms, entire organisms, from one biology to the other. But unless such a mechanism somehow evolved, there isn't any way to flip a cyanobacteria into its exact mirror. And so they would probably be able to eat each other, they would have the isomerases to eat each other's organics, but they wouldn't be able to flip into the mirror, so the mirror life would likely have evolved differently. And it's entirely possible that the mirror life never evolved the ability to get to Earth, even if terrestrial life is evolved from Martian life. So you could certainly have a scenario where there is mirror life on Mars. And so I will talk about that in a future video. I don't want this to be too long. So let's see, what should I just say? I just quickly touch on the things I'm going to talk about. So there's I'll go down, I'll show you the curiosity. That's the mirror life, mirror synthetic cell that I was telling you about, and which they actually expect to be able to make sometime in the future. And this is how everything, DNA, RNA, proteins, enzymes, everything is all just reflected. That'd be a big challenge. And so that's how they would sterilize it. And I'm going to talk about that. And this is to show that there could be life on Mars. Because if it wasn't, these are the curiosity brines. They're very cold, but Martian life might be able to survive there, and it could also form biofilms and retain it into the daytime. And so there could be, it is possible that there could be life on Mars, and this would also explain the Viking labeled release results, which are offset by a rather large amount. This is when they gave organics to the dust from the Mars surface, mixed it with some water, and then it evolved, and the organics was labelled with radioactivity, and so the sample, it evolved carbon dioxide or methane, we don't know which, during the day. And the interesting thing was that they produced most of it 2 hours, the peak was 2 hours after the peak of temperature. A few minutes, tens of minutes, could be explained by chemistry, but 2 hours is a very, very big offset to explain using chemistry. So that is one of the most suggestive reasons for thinking that actually maybe Viking did discover life. And then the other ones were confounded, the other experiments were confounded by the perchlorates, a very unusually reactive surface chemistry. If that is so, then there may actually be quite a high chance of returning life from Mars. So in that case, so I'll talk about my swan song biosphere hypothesis, which is the idea that just as Earth has Gaia keeps Earth very, very habitable, but it could be that on a Mars-like planet, and same photosynthesis that sequesters carbon dioxide into carbonates and into organics, then the same photosynthesis, into organics, then same photosynthesis on Mars, then over a very wide range of carbon dioxide output from the surface of the volcanoes over the last several billion years, then as soon as the surface it could be that volcanoes produce enough carbon dioxide to have liquid water on the surface and streams and everything all the way through to the present. But as soon as the liquid water forms, once you've got life there, then the photosynthetic life would proliferate, would take carbon dioxide out of the atmosphere. And so, over a very wide range of CO2 emission scenarios, you would get what we observe, which is that the water is very close to its triple point, and it's only barely habitable. And if this hypothesis is correct, then our Mars would be an example that is very common throughout the galaxy, that most Mars-like planets, if they produce a bit of carbon dioxide but not quite enough, then they will have, and if life evolves there, then it will equalize to the triple point of water, because as soon as it goes above that, it starts taking out carbon dioxide in large amounts. And from time to time you get a bit more carbon dioxide, the water starts to flow, but then immediately the life grows and pulls it back out again. So that is a way that Mars could have life all the way through to the present in what seems to be barely habitable. And this, in my view, I think this is quite a plausible hypothesis, but of course I would think that, since it was my idea. But it's not totally my idea. The other researchers have looked at this matter, that the atmosphere on Mars is very close to the triple point of water, and they suggest abiotic photosynthesis could be involved, along with sequestering of carbonates. And so the only thing I've really done is taken that research and said, what about biotic photosynthesis instead? And then that becomes swan song Gaia pretty much immediately. So that's where this idea of swan song Gaia has come from. It's quite a little leap from the research that there's already, just going to biotic photosynthesis and then thinking what would the consequences of that be. And even if you had quite a lot of carbon dioxide, then life would be very strong, a way of pulling it back down again until it's like that. So if that is what we find on Mars, then there may be quite a good chance of returning life in the sample if we do intelligent collecting of the sample. So I suggest the ESA fetch rover, we can't adapt the Perseverance anymore, but I suggest we adapt the ESA fetch rover to just take a little sample of the soil. Both Viking landers did get these labelled release results. So we want to find out what caused those results. Was it biology? Was it abiotic? Was it some strange chemistry? So and the Perseverance isn't going to do that. So I suggest the ESA-Fetch rover has an extra attachment which is going to be picking up these samples. I suggest it puts an extra, I've got an actual drawing of it down here, and that's the oxygen, strange oxygen signal, extra in summer and less in winter, and that just possibly could be, it would suggest it's very prolific life there, but it might be, you can bring up plausibility arguments for why these surface binds of curiosity, if they had biofilms in them, could actually be more productive of oxygen than, say, very dry regions in the Antarctica, which is what you would otherwise think would be there. So, and very interestingly, that the oxygen production goes down as the dust gets more opaque, which not a very strong correlation, but it's enough of a correlation to be quite suggestive. And that, of course, makes it look a bit like life. So, yes, I'm just going to show my thing down here. Have I got a picture? Yeah, that's how the scoop. You could have a little scoop thing like there, attached maybe to the outside of its arm. This is just a 3D visualization of it. But if you put a scoop on the outside there, maybe it could scoop a bit of dust onto an extra plate that you add on top of the sample receiving container. And that, I believe, would be of great astrobiological interest, and probably of much more interest to the astrobiologists than the samples. Because the samples, it's going to be very, very difficult to get early life that hasn't been deracemised by being warm for a long period of time, had the perchlorates and the hydrogen peroxide transform it. Then, if it's been on the surface at all, you've got all the ionizing radiation and so on. It's going to be very difficult to get non-degraded past life, especially since there's a constant inflow of organics from comets and meteorites. And we already have organics in our Martian meteorites that have multiple biosignatures, and even as we saw with that Mars meteorite, even what looked like tiny little cells in them, and yet we can't prove that that is life. So since Perseverance doesn't have the ability to detect life in situ, and we talk about Tissen meteorite as well, looks very much like life, but we can't prove it. So whatever we return there, will be of interest. It's be like returning some more Martian meteorites, but it's not going to resolve the central question in astrobiology. That's very, very unlikely. The astrobiologists have said in many papers, every single paper that I've seen by an astrobiologist on studying how we should look for life on Mars, say we have to send instruments there in situ. We have to go and look in situ. You have to narrow it in, home in on the regions of Mars where the life might be. Otherwise you could take back 1 billion samples, anyway, and you still would be missing because you're just not looking in the right places. Because and then after that 1 million samples you would find, oh well, that looks a little bit suggestive, maybe there was life somewhere near that one. And then you want to go back and then do more samples in that area. It's not feasible. Whereas if you're on the surface, you can go then, because say, oh, this is a good trail. Let's, like following the warmer, and you get closer to where the life is, and then you eventually find it. We can't do that with the samples returned, where you can't search for life in situ. So we're not likely to find. It's just a technology demo, really, of some astrobiological interest. But most of the organics returned, the organics is like ambiguous, and having the provenance, which is of great interest to geology, is not so interesting to astrobiology if you don't know if it's life or not. So it'll be of some interest, but it's not nearly as interesting as the geologists imagine it would be, because this mission is principally designed by geologists returning what they think might be of interest to astrobiology. It's not really that interesting, not worth the huge price. So it's just a technology demo. But if we could add this on the surface, I think that would certainly be of huge interest to astrobiology, whatever it finds, whether there's life there or not, because it would resolve this whole Viking labelled release thing, which has been, just nobody knows the answer, and say, was it life or wasn't it? Now we might know. If they don't do that, it could be another 2 decades before we know whether Viking discovered life in the 1970s. And the other thing would be of great interest, and I suggest that the ESA Fetch rover could also have a couple of these samples. And very sadly, the way it's done is so enclosed, the way they've done so automated and enclosed, that I was hoping that there would be a way that Perseverance could put one of those samples vertically on the surface to let dust fall into it, in order to catch some dust before putting a sample in. It doesn't seem possible. But ESA could do that. It could just take a sample, have a couple of spare sample tubes, and just take them out, put them on the surface, and leave them vertically through the dust storms for as long as it's there, and then put them back in, and that then would be a sample, and they could have ESA could put magnets in the bottom of the sample tubes to attract the dust, because we discovered that when you put anything magnetic on the or the entire sample tube magnetic, or whatever seems to work best, because that really does work. The dust piles up in little heaps on the surface whenever there's anything magnetic on the rover around those magnetic regions. So you could really attract lots of dust in, and that would be of great interest, because the dust would tell us whether, in the forward direction, it would help us understand whether Mars is safe for terrestrial life. If humans land in one part of Mars, will the contamination spread all over the surface of Mars in the dust? So we can look at the dust and see what the situation is for that. And then we also, though, if there's native Martian life, would almost certainly use the dust to spread. And perhaps, for instance, the biofilm might produce a sporulating body of some sort with which would cover itself with nanoparticles of iron oxide, and a little bit would be blown off in the dust, like a little dust particle. And when it gets into the vines again, then you'd already have not just one cell, but quite a few cells, a little bit of biofilm, which would then be able to retain water immediately through to the day, and so it would start growing quite quickly. So that's just sketching out an idea. And so, there could very well be spores of some sort, or something in the dust, and coated probably with iron oxide nanoparticles, in order to protect itself from UV and from perchlorates, especially the chlorates that would be activated by the UV, and so that there could well be life in the dust. So we should try and return some dust and find out, a remote chance of a spores. You'd probably have to collect quite a lot of dust, but you would have a chance. You'd learn a bit about the characteristics of the dust. So anyway, I suggest that. And we turn to Laplace plane. I think so what I was explaining. So the other thing I wanted to explain was that the best place for life on Mars are the Mars surface brines and dust. And we have no samples of Martian dust at present. We don't have any samples of lunar dust even, apart from what we got by going to the moon. So in the case of the moon, if there's something that lived in lunar dust and didn't even live in the rocks, we know that it's too dry, but just suppose that the moon's a little bit wetter and there's something lived in lunar dust, then it would be very unlikely it ever got transferred to Earth, never mind even though the moon is very, very close to Earth, because the dust would just it could blown off into space, and it would get sterilized very easily by the UV. But in the case of Mars, it's even worse. Then with Mars, then if any dust got it would go into the atmosphere. Going through the atmosphere, you have the fireball of leaving the Martian atmosphere, and then sterilized by the entire journey to Earth, and the UV and the ionizing radiation, even if it could get into space. But it so turns out that when a big asteroid hits Mars, it knocks off some of the rocks. It knocks it off from below 3 meters below the surface. So that part of Mars is believed to be sterile. It's too cold for life, almost everywhere. If you were to hit in just the right place, maybe where the RSLs are, depending on your hypothesis as to how they formed, then maybe there's life below the surface there, in a kind of hotspot of some sort. But we haven't yet discovered any hotspots on Mars. We know they exist, or have existed in the past because it's still geologically active, but we don't know of any just now, so they aren't very common. So it's not easy for life to be transferred from present-day Mars to Earth, and the surface brine and dust have never been transferred. So there we have it, that Mars, we certainly don't know that Mars is sterile. All all the sample return studies have looked at that and said we don't know it's sterile. They also say we don't know if whatever life there is on Mars has been transferred to Earth. And I think this it reinforces that when you look at this particular case of the Curiosity brines and the idea of a biosphere film that could make it more habitable. That's Nilton Renault's idea. He wrote the big long paper, wrote the book on water on Mars and surface water on Mars especially. And he thinks that life could form a biofilm that would retain water into the daytime. He just as a speculative remark, not a paper. In one of his interviews, he mentions this suggestion. So I think there could be life on Mars. Hopefully I've made a suggestion as to why really quite a high probability even. There could even be quite a high probability of surface life on Mars. It could easily be something hazardous like mirror life. And in that situation we absolutely have to know what we're going to do there. And for any of you who are space colonization enthusiasts, then in some ways this, and I am still a moon space colonization enthusiast. I used to think, oh great to have humans go to Mars. I never even thought about all this. Just a little gap, I didn't make the connection. That I knew about you need planet protection, you kind of get human-centric and think, oh well, if humans are there, they don't need to be any protection, we're fine, even though robots need to be sterilized. That's a kind of sort of human anthropocism. We kind of think humans are okay. We can't possibly bring contamination to Mars, although our robots do, which of course is the other way around. That humans are the issue and robots can be sterilized. And so, so if it turns out, it's certainly possible, if there's mirror life on Mars, then there could be the situation that we simply can never safely return unsterilized samples from Mars to Earth. That is a possible situation. So, and so Mars colonization enthusiasts, I think, need to be prepared for that. We can't know in advance. It is a possible situation, and we can only find out by researching and finding out what is there. And in that situation, actually, I think it would be very good for space colonization, because you would get a very strong incentive to have humans living in orbit around Mars, studying the surface by telepresence. They would never be able to go down there, but they would be like exploring the seabed, but with avatars and greatly improved technology, robotic avatars on the surface. So they would be there exploring Mars from orbit, and this would unlock billions and billions of dollars of funding to get people to go up there to go and search with very low telepresence. Anyway, so that would be really quite an incentive for Mars settlement, I would say, but Mars settlement in orbit and of the Martian moons, if it had mirror life there, or some other form of life that's very interesting but hazardous. So that's that. And then just to say, so the idea is to return it to the past plane, which is perhaps 10,000. So did I mention that? It's basically a swing plane. If we had a ring system like Saturn, then the rings wouldn't orbit in geostationary Earth orbit. They would orbit at about 7 degrees to that orbit, at that geostationary Earth orbit. And it just so happens, it turns out someone has done the study. They were looking at safe places to dispose of geostationary Earth orbit satellites, GEO satellites, and they found that if you put them into the past plane, and then they explode or bits get broken off or something, they're not going to go back to equatorial orbit. They're constrained into the past plane, and then you would think, but of course the past plane surely intersects with geostationary Earth orbit, because the planes intersect at least. But it turns out that if you have something released from the past plane, the ring plane of Earth, then it just doesn't go back into the geostationary Earth orbit. And so if you put it in the right place. And so I suggest that we return it to 10,000 kilometers above GEO, or so, or more, 20,000, 30,000, whatever works best, at an angle of inclination of 7 degrees. And then we study it above GEO, and the way we do that is we are sending missions to the satellite instead of to Mars. And the numerous miniature instruments that astrobiologists have developed, and they'd be very eager to send them up, at least to the satellite, even if they can't send them to Mars yet, and they can test them there. And they can find all sorts of interesting things about this sample, and then we can design new instruments and send them up there, just to the GEO, and then telerobotic exploration with almost no latency, like exploring Mars but with almost no latency. So that I think would be no risk to Earth's biosphere. I think that'd be a very exciting and interesting mission. Now, the very interesting thing here is that you would still sterilize the sample, but you would sterilize bits of it, and then return it to Earth's surface. And you do that straight away. Then it turns out you can sterilize it in such ways to have no impact on the geology, apart from a slight discoloration of quartz, turns from clear to brown, and halite crystals, salt crystals to blue from clear. Apart from that, hydration isn't changed, the interspatial distances aren't changed. The radioisotope dating isn't changed. It doesn't trigger radioisotopes to split. And that's using gamma rays. You can also use tuned X-rays. So that is a way that you could do that. And so you return the first sterilized bits of it, and then you'd look at those, and then you'd see, is this does this look as if what we have up there is biological? So that would also be preserved if you use just the right dose of sterilization. Then you would still you'd recognize the shape of life, you'd recognize complex molecules like chlorophyll, you'd recognize chirality signature. And so if there's present-day life there, you would recognize there had been life there. And so I suggest that would be great. That would be, if you did that, then you'd say, okay, we're not going to sterilize the entire thing. But then if you think it's not interesting, then you just sterilize the whole thing and return it. And then in that case, it just becomes like the returning of a sterile sample all the way. But you have this pause above GEO for a few months and sterilizing part of it and examining it, and then you say, okay, we're going to sterilize the whole thing and just bring it back sterile. But then if, on the other hand, we did discover, for instance, that this pile of dust, imagine this pile of soil and pile of dirt, and if we managed to return that, and then we say, oh, there is life in there, and oh, it's mirror life. In that situation, first of all, if we knew for certainty there was mirror life there, I'm pretty sure that nobody would even think about trying to return that to a terrestrial laboratory. The only reason that they consider that is because they think there's a very small chance of there being life there, and they're just not looking at things that can have large-scale effects. So we find this mirror life there, then we would always do it in the forward direction, always send instruments up there, not bring them back, and then bring back only sterilized material. And that could be the nucleus of a laboratory that could then be used to receive samples of life from throughout the solar system, from Europa, Enceladus, and Ceres, if there's life in Ceres, and anywhere in the solar system, even if there's life in comets, which some people think is a very remote possibility. Wherever there's life, we could then return it that gradually in growing facility. And we could use Elon Musk, his great big spaceships that he's building, his Starline or whatever he calls them. And he thinks that they can actually be very low cost, and they can be able to buy them second hand later on, once he's got a production chain up for them. Well, a second hand one of those would be a great nucleus to start one of these facilities, to add to the facility, and then you could have a huge amount of space inside to do all this research. All controlled by avatars from Earth, and no astronaut ever goes near there, because you don't want to return mirror life to Earth. Because if you've got mirror life to a human microbiome, then it would be, and suppose it can survive in your human microbiome, then how are you ever going to how can you quarantine that? It seems that, I think, is a very clear example of why quarantine just doesn't work as a way of protecting a planet. Unless you know, but quarantine does work if you know what it is you're returning. That's the one thing I forgot to say. So if you know what you're returning, then quarantine does work, can work, if you know what you're quarantining against. and if you know what form of life there is on Mars. So so I've looked at because of the whole legal process thing, I've looked at the case where what's on Mars is harmful to Earth. But you could also have something on Mars that is beneficial to Earth. For instance, the entire domain of the Archaea. Then there's nothing there that's pathogenic to humans. It's quite remarkable. An entire domain of life with nothing there that is seriously pathogenic to humans. It might be involved in tooth decay, and there's a few things like that. But so it is possible, but we need to understand the biology very well first. It's possible you could have an entire domain of life from Mars, return it to Earth, and it's completely harmless and even beneficial to the terrestrial biosphere. So I'm not saying that's not possible, it is possible. But you need to be very, very sure that that's what you've got there. And you can bring up other scenarios like that. So if we look at this very carefully, maybe later on when we have a great deal of understanding of what's there, if there is life there, and of course if it's sterile there, then that's another situation. If it's sterile, you still want to be careful in the forward direction, because this would be our only opportunity to study what happens to a Mars-like planet which hasn't got life in it. So even if Mars is completely sterile, maybe eventually we send humans there, but I think even then we should hold off and study it first from orbit, and then think about what will the effect be of terrestrial life on this planet. And meanwhile, we've got the Moon, and the Moon is a fabulous place for. Really, it gets very. People say, "Oh, we've been to the Moon. Been there, done it. Now we go to Mars. When we've got to Mars, we've been there, done it. Then we go off to Europa, been there, done it. Gone off to Titan, been there, done it. Now we've contaminated all the interesting places on Mars in our solar system with life. So let's go and look for another solar system, another star system, and then we've been there and done it." We don't want the been there, done it attitude. So the Moon is incredibly interesting, and the possible ice of the caves at the poles. It could have organics from meteorites. It could have meteorites from early Earth. It could be early life on Mars, on the Moon, in the polar ice. We could be able to actually find just actually organics preserved from early life. Bits of not arachnids, ammonites, bits of ammonites in lunar ice, for instance. Actually, the actual bits with the actual DNA. And could actually even be preserved at liquid nitrogen temperatures ever since the time of the dinosaurs. It's not impossible. You could find DNA from the time of the dinosaurs up on the Moon. And then the huge lunar caves, and I can wax lyrical for a long time about the Moon. It's a very interesting place. And I think we should be we should start with the Moon. It's far closer. You could get medevac back to Earth in a couple of days, rather than 6 months. It's just such a big leap from having medevac of 2 days to having a medevac of 6 months or a year or more, depending on the timing. If you've got something serious going wrong. And then the idea of an Apollo 13 kind of mission heading off from Mars, where there's an explosion on its way out, just as it leaves Earth orbit, and it just doesn't have the delta V to come back again. That would be so tragic. And the Moon is also, it's in terms of space settlement, then you have the possibility of commercial use, you have possibility of tourism, but you won't have on Mars for a long time. Like Chris Hadfield said, we're going to go on to the Moon for a generation before we're heading off to Mars, and I think that is the best way to think about it myself. And just be ambitious for the rest of the solar system, but let's look at the Moon, and we've got so much interesting there. And so I do hope that if for those of you who are space settlement enthusiasts, please, this is not, this could actually good, you want to be behind all this. We want to be going together and not fighting each other. And so I hope that we can have a situation of a shared vision, instead of like the space colonists going one way and the scientists going the other way, and the scientists are appalled by what these space colonists do, and the space colonists being appalled by all the restrictions that scientists want to put on them. But I hope instead that we can be working together, all of us, and that we can see a shared objective and a shared reason, and we all care about the Earth. And if you just take this seriously, that this is a real possibility, if I haven't made loads of mistakes, and I don't see how I have, then this means that we do have a responsibility for the Earth, and we do have to be careful about it, because it is so precious. There's nothing even remotely like Earth in our solar system, and there's nowhere else where human beings could survive without at least quite a lot of gear. I think the best place in terms of minimal gear, apart from the Earth, would be Titan, actually, but again that's a subject for another talk. So anyway, that's it. This went on a bit longer than I expected. I was going to do a whole lot of small ones, maybe I still will, but I think I'll upload this one, and then I'll think about what I'll do in the future. I will put the link to the actual paper in the video description. Also, for those of you who are less technical about all this, and I have my OK to Touch Mars book. So that's just a general planet protection book that you might find interesting. Free to read online, and hopefully it's got some quite stimulating, interesting stuff in it. And but yes, I very much welcome any feedback on this whole thing, and any thoughts, any comments, anything I got wrong, anything I missed, and so on. And thanks for watching this. If you've got this far, then I hope you had enjoyed the talk. Presumably you did, if you have stayed the course to the end here. And thanks for listening.